So again, welcome to the AYA session for everyone who's here and everyone who's joining us online. We've got an action-packed lineup for you today. First, we'll start with several study updates for about the first half an hour. And then we've got special visitor Brad Pollock from UC Davis to talk about the new AYA COVID vaccine study. And another special visitor, Wendy Allen Rhodes from the Mayo Clinic, to review the recent NCTN sarcoma protocols. And then for the last half hour or so, we'll talk about AYA education. Adam Duvall is here from the University of Chicago across town to talk about gaps in AYA oncology education during fellowship, comparing medical oncology and pediatric oncology. And following that and related is a video that Jonathan Summers and colleagues and I developed in a recent PCORI grant that I did. And this is some patient viewpoints from how they want to be treated in their oncology care. So it's kind of the flip side of what he's studying directly from the mouths of patients. So first we're going to start with Alex Herrera giving an update on S1826. And he, I believe, is in the airport lounge getting ready to fly here to Chicago from Seattle. So do we need to unmute Dr. Herrera?
I don't see him on.
He's not on? Interesting. Okay. We could then come back to him. Can you guys look for him to pop on? Do you have Dr. Nappy?
Yeah, I'm here. I'm here, Becky.
All right, let's go to yours. Thank you. Oops, there you go. Okay, I'll advance for you, Lucia.
Okay, thank you very much, Becky. So I'm going to give you the updates about S1823, which is a clinical trial of microRNA 371 in patients with early stage germ cell tumors. I'm the co-chair of this study with Dr. Craig Nichols. Next slide. So just a little bit of background about microRNA 371. This is a very small RNA fragment. It's only 20 to 23 bases pair. And it's fascinating because it's extremely highly expressed in germ cell tumors, both seminoma and non-seminoma germ cell tumor. Oops, can you go back?
Sorry.
It's extremely specific for viable germ cell tumors. There are no other disease or conditions that express these microRNA except for physiologic pregnancies. It is not expressed by teratoma, so that's the only limitation of this test, especially in patients with germ cell tumors, non-seminoma germ cell tumors. And it is abundantly secreted in the bloodstream, so that a very small amount of blood can be used to detect microRNA 371. We can use either serum, or in my lab, we use plasma to detect these microRNA. It is expressed in both pediatric and adult germ cell tumors. So as you can see here in this heat map at the bottom of the slide, the tissue with no germ cell tumor teratoma in it do not express these microRNA clusters, so it's in blue. And in both pediatrics and adult germ cell tumor tissue, there is high expression of these clusters of microRNAs. It also rapidly decreases after orchiectomy or after active chemotherapy. So the half-life is much shorter than what we currently have for our serum tumor markers. Next, please. So the microRNA 371 expression directly correlates with the tumor burden. So the higher is the tumor burden, therefore, the more advanced is the stage of germ cell tumor, the higher is the expression. and can be detected in pre-surgery, so prior to the surgery in patients with the clinical stage 1, and disappears after the orchiectomy if there is no residual disease. And it's detectable in most of the patients with metastatic germ cell tumor. The other difference between this microRNA and other circulating RNAs or circulating tumor DNA is that it's extremely stable. So it's not degraded by circulating RNases. Therefore, it can be stored for years in the right condition in the plasma or serum. Next, please. It can be detected in a very small amount of blood, as I mentioned, and what we currently use for the quantification is a real-time PCR, which is a very commercially available, I would say, assay, not particularly complicated. Next, please. So this is the half-life of microRNA 371. And as I mentioned, the half-life is about 18 hours, which is much shorter than what we have for beta-HCG, which is 24-48 hours, and alpha-fetoprotein, which is about 7 days. Next, please. Next. So based on this background, we did publish a pilot study on JCO in 2019, where we confirmed a very high accuracy of microRNA 371 compared to CT scan and tumor markers in detecting active germ cell tumor across the spectrum of germ cell malignancies. So the sensitivity in this pilot study was 96%. The specificity was 100% because it didn't tell any false positive results. And the accuracy overall was 0.97 compared to 0.77 of CT scan and between 0.60 and 75 of the tumor marker. So it seems to appear that across these, again, very heterogeneous kind of populations of patients with germ cell tumors, this microRNA was much more accurate. Next, please. So in terms of using this biomarker in the clinical setting, we are at the clinical validity and analytical validity stage, so we still need a clinical utility validation, and that is why we decide to design S1823. So this is the schema of the trial. Essentially, it's a very simple study. Patient with the clinical stage 1, stage 2 in the negative tumor markers, or also more advanced germ cell tumor were eligible for this study. They needed to consent to have blood collection at a pre-specified time point. Go back, please. The pre-specified time points were decided according to the theoretical risk of tumor relapse. So we had 3 groups: low, moderate, and high risk. The blood collected in the Streck tubes is shipped to nationwide for biorepository and plasma isolation, and then I receive the aliquots of blood samples in batches to Vancouver for the microRNA 371 analysis. So there is no real-time results of this microRNA 371 available, and the results of microRNA does not inform clinical decision. Importantly, in order to define the primary endpoint, we also collect blood during follow-up and the time of relapse for the patients who are going to have a relapse of the disease. Next, please. So this is the schedule of the blood collection for patients with the low risk, moderate risk, and the next, please. High-risk patients. So the high-risk patients are patients with metastatic disease. So in this case, we only collect blood prior to the primary treatment and post-treatment, and eventually post-surgery if the patient also needs surgery, while the schedule for the blood collection is a little bit more intense for the patients with moderate risk. and it's every 6 months for the patients with low risk. So we are amending the protocol also for patients with moderate risk to have less blood sample collected, but at the present time, this time it's every 3 months for these patients with moderate risk and every 6 months patients with low risk, for 2 years if the patient does not have a relapse of the disease. Next, please. So the eligibility is extremely flexible. Every patient with newly diagnosed germ cell tumor was eligible for this study. However, we have reached the pre-specified number of high-risk patients, so the patients with metastatic disease are no longer eligible for this study. So we are currently only accruing patients with low and moderate risk. Next, please. And the primary endpoint of the study is to establish the positive predictive value of these microRNA in predicting active germ cell malignancy in early stage real-world germ cell tumor, and to establish by how much time, if at all, the microRNA 371 expression precedes the identification of tumor relapse in patients with germ cell malignancy. The secondary endpoint is to establish a clinical annotated biobank for future translational studies. So we are also collecting germline DNA as well as plasma for eventually extraction of circulating tumor DNA. Next, please. So in our statistical design, we target 50 relapse in seminoma and 100 in non-seminoma. And based on this assumption, we expect to recruit 956 patients in 2 to 3 years. And the period of observation is 2 years, so the patients are not going to relapse. And we plan the interim analysis after the first 40 relapses. Next, please. So the study was activated in the US first and then in Canada. In the US in June 2020, and in Canada we enrolled the first patients in December 2020. Next. And as last week, we have enrolled 593 patients. The 956 patients we target to enroll. As I mentioned, the high-risk cohort is now closed. And we have most of our patients with lower risk and 103 patients with moderate risk germ cell tumor. We haven't had a lot of relapse so far, but the median follow-up is still short. So it's everything is within what we expected for this study, I would say. Next, please. So we are working, as mentioned, on a revision of the protocol to reduce the number of samples for patients with moderate risk, and also to make easier the recruitment of the patients. So we are modifying some of the eligibility criteria in order to have like easier access to the study, without obviously affecting the statistical design and all the other most important factor for the study. We are also extending the time from diagnosis to enrollment. Currently it's 42 days, but we had some feedbacks from the sites that it's probably too tight in certain circumstances, and therefore we want to extend it to 60 days. So in the revised protocol, that will be also changed, that hopefully will facilitate enrollment. Next, please. In terms of funds, we did receive an R01 that was transformed in R37 from NIH. So these will fund the analytics cost of this clinical trial. And I also received a grant from the US Department of Defense in 2022, was a Career Development Award that will support my lab and this work in microRNA germ cell tumors. Next, please. And I think that's my last slide. Thank you.
Thank you so much, Dr. Nappi. Questions for her? You can either come up to the microphone here, or if you're listening remotely, you can write them on the chat and I will share them with the group. What will the funding of your new grants have you working on? Is it all related to this clinical trial, or will it be follow-up after you have analyzed this data?
So for the Department of Defense, is related to this grant and to some projects that we have in collaboration with Dr. Management in LA and some military collaborators in the US. So it's possibly applicable to this study, but not for the primary endpoint. So most likely later on.
Great. And your modifications, I don't know if it's possible to tell this, but are any of them, do you think, related to the care of AYAs as a group from the feedback you've gotten from the sites? Or is it just more related to the follow-up of germ cell tumor patients in general, such as the extending the time for the initial enrollment?
Yeah, I wouldn't say that that's related to AYA specifically issues. It's more related to giving it more flexibility in order to enroll the patients, like, for example, the time from diagnosis to enrollment, or also making like tumor markers, post-orchiectomy, not an eligibility criteria. So we are not affecting the study, obviously, but we are making things easier for the sites in order to enroll more patients.
All right, great. Any other questions for Dr. Nappi? No. Do we have Dr. Herrera on the line yet?
Not yet, no.
Huh. All right. We'll just move forward, and you can tell me if he pops on, and we can just incorporate him in whenever he has time. He was catching a flight, so I hope he didn't. I hope he estimated his time properly. What happened to our slides up here? Okay. So next, no, mine's not enrolling, or mine's not advancing. So I'm going to talk about John Saltzman's study, which is the quality of life AYA PRO study that he presented on at the spring meeting. The accrual for that overall is going really well. So I'll share a few follow-up slides with you once we have them. I can advance them. Thanks. All right. So this is EAQ 202, Improving Adolescent and Young Adult Self-Reported Data in ECOG Akron Trials. It's an NCTN-wide study run through the NCORE network. John Saltzman is the PI, and we have several study champions throughout the NCTN, and I'm pleased that SWOG is one of those. So the primary objective of this trial is to evaluate the feasibility and acceptability of completing PROs, patient-reported outcomes, among AYAs. And in this study, they're randomized to either have a core battery of PROs shared with them, and then they answer those questions, or they can choose the topics that they're interested in at each time point, and they can fill those out. And the exploratory objectives, therefore, are to describe the AYA's health-related quality of life priorities and the trajectories through their cancer treatment. And it's a novel and pretty unique way to do this that will be very interesting. Second, they are evaluating access to and utilization of both psychosocial and financial resources using these PROs. And then they will summarize AYA's preferences for how their PRO data should be shared with them and or their providers. And here's the study schema. They will be getting surveys of either the choice PRO in the top row, or the control arm is fixed PROs at these time points: baseline, 1 month, 3 months, 6 months, and then at 12 months. Oops, Yeah. And again, it's going out through NCORE. It's got a nice reimbursement of, I think, about $1,000 per patient. And the responsibility of the registering site is only to screen and consent the patient, register them, and then see them as clinically indicated for their cancer care. And then the study team will actually deploy all of the PROs via EasyPro, an electronic entry system, and then monitor all the follow-up and send all of the texts and emails and calls promoting adherence for the patient. So it should be a very feasible study for sites to open. Accruals are going very well overall. There are now more than 295 sites with IRB approval for these, and they've so far enrolled 275 patients with lots of activity through the spring and summer. They have already met the accrual goals for breast cancer and are looking for patients with leukemias and lymphomas, colorectal, germ cell tumors, and other cancers. These are all early stage, good prognosis cancers as opposed to patients with metastatic disease. Of note, they are wanting 47% male population and currently are less than that at about a third, and so also seeking male patients. And John Salzman is the study chair. This is his email for any questions, and you can also direct them to me here within SWOG. Thank you. Questions or comments on that study? All right. So now I'd like to introduce Dr. Brad Pollack from UC Davis, who will be talking about the vaccine study that he's developed.
Thank you, Becky. So I'll just give you a little bit of background. I'm not new to this AYA field. I'm actually the PI of the Children's Oncology Group NCORE research base, and I'm an epidemiologist and biostatistician. I have been involved with AYA since I think we had our first meeting in 1997. Archie Blyer and Ronnie Barr and several others were there to organize this. This is before the pediatric groups actually came together. So it's been wonderful to see this kind of develop as a subdiscipline in of its own right. And one other little digression here. So I've been at UC. I actually I was in San Antonio for 13 years. Dr. Coltman recruited me there as the Cancer Center biostatistician. And so I used to go to SWOG meetings pretty regularly. And I think I gave a talk on AYA stuff probably 15 years ago in Kathy Albane's special populations group. So it's nice to see that we've got a permanent committee here. The other little digression is that while I'm a cancer epidemiologist primarily, I also chair a department of public health sciences at UC Davis, and got roped into running the University of California system-wide COVID-19 public health work group for 2.5 years. So I got dragged into COVID and infectious disease epidemiology in ways I never imagined. And I'll just finalize that by saying that I also ran a 2-year project in Davis, this Healthy Davis Together project, which is just finished up. We've gotten a lot of national attention, but we had a very successful community intervention with COVID by doing everything you could possibly do at a cost of about $40 million to keep COVID out of the community, keep the rates down. So I've been kind of juggling these two worlds for the last 2.5 years. And I guess about a little over a year ago, we were talking a little bit about, there was a registry study we had in pediatrics looking at cancer patients that got COVID. And so that we were part of that consortium. But the issues really came up about vaccination and what was going on. And of course, it's not a new issue. So Imad and myself and several of my colleagues, San Antonio, from Philadelphia, other places, we were having some discussions about trying to put together a study to look at this. And meanwhile, the NCI was very interested in this. They have their SeroNet group intramurally that we're looking at developing some opportunities to look at vaccine and patients that are on chemotherapy. So we put together this concept. It went from concept to activation in 7 months. So Dr. McCaskill-Stevens has used us as the poster children for the NCTN groups and the NCORE research bases about how quickly you can get things done. But we are looking to expand this out in terms of partners, and we had this in mind all along. So I'm going to talk a little bit about the protocol, which is currently active. But the background here is pretty straightforward. Obviously, cancer patients are at greater risk for having severe COVID infections. We've seen that. That's pretty well documented, and that's true in pediatrics. Immunosuppression from the underlying cancer itself, as well as cancer-directed therapies, play a role more likely in terms of modulating the vaccine effects. They certainly modulate the severity of the infection itself. And so we know that's important. And most importantly, we have just very limited serologic data available on the response to the vaccines under different conditions of different types of therapy, including those that tend to be more immunosuppressive. So this is kind of a statement of facts there, and it hasn't stopped other groups. And the NCCN has a standing committee that has put together some guidelines now for vaccinations in cancer patients for COVID vaccines. This actually gets updated. So their primary recommendation is, yes, give an additional dose. That's kind of the gross recommendation here, but they've been working away, and they now have a version 7 of their recommendations here, and they break this down by heme malignancies, solid tumors, and so on and so forth. And I will say that most of this is really based on expert consensus. They got the group together. I know several people are on the committee said, yeah, this sounds right. It's not really evidence-based because we don't have the data to support that. So this is probably the best paper I've seen. This came out, I think in April this year. Fendler over in Europe. But these were the take-home messages. This article is a really good review. Again, it's a little bit dated. The information they had available to them at the time was up to Delta. They had some Delta information for the Delta variant of concern. But these are just take-home messages here that, they know that administering vaccines with the boosts is safe and clinically effective. They're seeing, positive effects from that. To date, there's no reliable correlate of protection defining clinical efficacy or immune responses from these vaccinations. We don't have the laboratory measures. And in that review article, they go through what people have done, looking at antibody binding affinity, many other measures here, but we just don't have the large database in this population. And where we do have the data, it's been in these massive population-based studies of 20, 30,000 individuals where there happen to be some cancer patients. And oh, by the way, let's describe their experience. So not quite the angle that we're coming at it from. We know that factors such as advanced age, certainly heme malignancies, other clinical characteristics, of course, and then, of course, the treatment-related factors themselves are likely to modulate the effect of the vaccine and as well as interact with the cancer therapy. We don't know. And of course, there are recommendations here where the future research needs to determine the necessity of these booster regimens. And let me just tell you, go back 13 months ago when we were sitting down trying to plan the study out, boosters weren't even a regular thing quite yet, let alone multiple boosters, let alone bivalent boosters. So this has been a moving target. We still had plenty of people getting J&J, and we didn't have people, and we have some of the other sites. We have all the Canadian institutions that are part of our group. I think they did not do AstraZeneca. I think they did just the messenger RNAs, but we have some European sites as well where they were getting other vaccines. No Sputnik 5 to my knowledge. So obviously we're left with little information about what's going on here. That was our motivation. So we wanted to look specifically at the clinical effectiveness, I should say, of COVID-19 vaccinations in children and adolescents and young adults during chemotherapy. So that's the primary aim here, is to characterize the immune response following vaccination in this population, and do this again close to preferably when they're going to start their cancer therapy, but we even allowed for some slop there in terms of people that have already started the cancer therapy. But they have to be, you'll see in the eligibility, vaccine naive, which is a bigger issue for adults, but believe it or not, even today, 25% of kids are vaccinated, 75% are not. So we're not having an accrual problem because of that eligibility criterion, but even in the adult population, it's kind of a mixed bag. Okay, so there's some exploratory aims here. One is to look at symptomatic breakthrough infections. We were not going to do the kind of monitoring, weekly PCR monitoring in this cohort. That would have been just impossible. So we were going to confine ourselves to looking at reported COVID infections, really. And I think that's still going to be a weak need endpoint now, since Omicron came along. We're seeing less severe infections. Who knows what's getting reported and so on. So that may be a little bit of a tougher aim right now. We want to look at the durability of the immune response to the vaccines over a 2-year period of time, and look at some of the longer term impacts here, including some subsequent COVID-19 related serious illness, long hauler syndrome, and so on. And then I have some other things here. By doing this study, we're hoping to learn a little bit more about the biology of what's going on that may help drive dosing and timing. Again, it's going to be a challenge. I'll talk about that in a second here, but looking at some of these attributes of the immune response and so on. And then most importantly, this study is going to relies on biobank. We're using our biobank headquarters in Columbus, Ohio, but we're having aliquots there because we know what's going on is, even since the time we started this concept here, the serologic assay technology has gotten better, more efficient. It's evolving so quickly. So it's going to be really important to be able to bank this material so that when we're looking maybe 2.5 years down the line, we'll be able to go in with some of these aliquots and do some assays that either don't exist today or really are still fairly imprecise. Okay, so, I'm sorry, skip the slide. Let's see about that. Okay, got a hair trigger here. So the eligibility, it's 6 months old up to 36. There has to be the plan to receive a vaccine, and that's important. This is not a vaccine trial. We were not trying to coerce people, so we made this very clear in all of our discussions that the decision to vaccinate has to have been made prior to considering enrollment onto this study. It's not part of this study itself. It's an incredibly important question, who's getting on and why aren't they getting on, but that's beyond the scope of what we're doing here. They have to have a confirmed cancer diagnosis, of course, and they have to be treated in a North American institution. The reason for this, again, was the viability of specimens being shipped on. We have New Zealand, Australia, Groningen, the Netherlands, and we were just concerned about the viability of some of these assays that we want to do. And then it has to be, of course, feasible to collect these specimens prior to the initial vaccine dose. And today we had somebody that was enrolled, collected the pre-vaccine doses, and they wanted to find out, it's supposed to be done within 5 days, and they actually start, and they need an extra day there. So we're trying to figure out what we're going to do to adjudicate that slight deviation there. But this is at the baseline level. Okay, so we had to sort of set up, sort of a straw man for determining whether there's an effect or not. So we were using a very dumb proportion of folks that basically had a humoral response to this by looking at SARS-CoV-2 specific antibodies here by day 30 post a vaccine administration. Now we have several time points built into this. I'll show you in just a second here. The incidence of breakthrough cases, we would look at that with an incidence density approach, but again, I think there's going to be a vast underreporting there, and so I'm not sure we're going to get good data for that one. But this is really a straw man, in my opinion, for what we're going to do for the assays here. So this is the schema in terms of time points. We start at baseline. There's a 30-day period then from any vaccine, and then there's vaccine administration. 30 days post each dose of vaccine, we are collecting blood, collecting specimens for this. And there's a schedule laid out there that there'll be 3 months, 6 months, this is not after each dose, but basically an interval no more than those periods of time there out to 24 months. And looking at this with a little bit more specificity, this kind of lays out the timelines here and the protocol itself. But we have allowed for some slop, believe it or not, because you can't assure that you're going to get a patient in every day when they come up with their 30-day average and so on. So we've got some tolerances built in for that to allow flexibility to coincide with their schedule for getting the regular treatment. And this is actually, believe it or not, we're still in discussions with SeroNet, and SeroNet, the NCI has contracted out to Quest Diagnostics. So Quest is actually doing most of these assays. And so, believe it or not, even Quest has some heterogeneity within their corporation about which labs are using for which assays. They have 2 main labs, Chantilly, and there's one in San Juan Capistrano. And we're going to divide the aliquots, believe it or not, to those 2 labs based on where they're doing, sort of prefer to do this batch type processing. But these are still in development. You can see there's a wide range of things here. Key thing is we're doing neutralizing antibody, which is a very important part of this, and neutralizing antibody specific to the variants of concern. So that's probably our best measure, although kind of the trickiest and most expensive to do. But there's other serologic quantitation that's going to be done here. And some of these are just qualitative, yes, no, that sort of thing. So again, this technology is evolving very quickly. We know that even in a year from now, you're going to see, essentially machines and assays that are going to be much, much better than this. So that's why the biobanking is really important. But again, all right. So just a little bit on the sample size, the planning for this thing. We just to look at backgrounds here. We did look at some of the adult studies where there was evidence of antibodies to the vaccines. And these were present in almost every cancer patient that looked at, certainly in the solid tumor patients, but even for the stem cell transplant patients, there was evidence of measurable antibody. As I mentioned, for the protocol, we used a straw man of 70% vaccine response. That's a yes or no response, and that's a very, very gross thing. And we don't plan to use that as our primary efficacy measures when we're doing the analysis, but just to get some ballpark sample size. So we estimated using this criterion, we'd need about with inflation for about a 20% inevaluable rate, about 532 patients. And that's, again, to look at the primary endpoint for the seroprevalence. But again, we'll have more sensitivity for some of the more quantitative measures as well. Okay, so I am having a problem with this clicker here. Is that it? Okay, there we go. Nope.
Very enthusiastic.
Becky, I think it is, and I have a very heavy hand. You want to go advance the next slide? Let's see what that is. Computer will work, right? Oh, it doesn't work. Okay, I'll do it here. Sample size limitations. Okay, so just full disclosure here. The major limitation is the sample size. This is a prospective cohort study, an observational study. Even with 500, if we got all 500 subjects that were evaluable, our ability to really tease apart the heterogeneity reflected both in the vaccination process as well as the cancer therapy is going to be very, very difficult. And just think about this. Which vaccines did they get? What was their dose schedule? Are they going to be boosted? What's the schedule for boosting? Now we have bivalent mRNA vaccines. And so, we're going to have to build this in. That's just source of heterogeneity to try to pull together some trends. But we're going to try to do that. And then with cancer therapy, again, we have some very enthusiastic folks on the study team. We want to expand that out, but they're all going to be doing a lot of sort of ad hoc coding of things like degree of immunosuppression, and it's going to be kind of a wild ride there. We certainly can divide up into heme and solid tumors, but we want to do that with much more specificity. So that's going to rely a lot on our clinical judgment and being able to put that together. But I think we'll be able to. These are primarily protocol treated patients for us. And then of course we've got the other problem that the virus doesn't want to stand still, right? We've had this evolution of variants of concern, known variants of concern, subvariants, who knows what's coming next. And then even for the vaccines, I think a lot of us felt all along that targeting just the spike protein, good thing to do, but hey, we wouldn't do this in oncology. We need multiple targets. So there are other proteins that could have been targeted. I know there's some vaccine research going on that level here, but we may come up with vaccines that can provide a much more robust protection over time. So we don't have that now, but I expect that during the life of this study, we might see some prototypes of that. And of course, there's a lot of other heterogeneity from other factors there. Okay, so the study opened in April. We have accrued, and the update date was as of the 16th. We have 37 institutions that have opened it in COG, 42 enrolled from 12 sites so far. And we're just doing a big push right now in COG. This is the accrual pattern. That looks pretty good for us. Even building in the Abrams rule there, where they gave us a free ride for 6 months. But we're moving along. And this is just a list of the diagnoses of the 37 odd patients that are enrolled so far. I don't know if that adds up correctly, but as you can see, we do a lot of leukemia, so that's our predominant numbers there, with a scattering of some other solid tumors and lymphomas. And again, a male-female ratio, it looks pretty close to reality. So with that, I just wanted to say that we really had envisioned trying to do this as a study that we'd open up, and we'd love to have the adult NCDN research bases do this. That was really, the NCI is pushing us to do that also. It's very much in keeping with my philosophy. And so I'm here to talk about that. And Becky expressed a lot of interest also. So happy to answer some questions. I'm sorry I went a little bit over my time, but.
No problem, you're good.
You made me one speaker short anyway today, so, okay.
Alex said he logged on.
Do I go up for?
Yes, please.
Just so the other folks can hear you online,
Yeah.
yeah.
It's a really interesting study. I work in a lymphoma in my lymphoma clinic, and I saw that you guys had a lot of leukemia patients, which makes sense because there's so many children that have that disease. And I was just wondering, you know, if you would, because of the agents that the lymphoma patients get versus solid tumors, what type of impact you're planning on seeing, because we've had patients come in specifically who have like CLL and who are getting rituximab, and they get their booster 2 weeks later, they get the antibody test and it's 0.
Yeah,
So-
so that Fedler article that I put up there, they did a phenomenal job. In that article, they went through and they separated out the malignancy types and biologic versus the normal cytoreductive therapy. So there actually is, there's some preliminary data in some places, and it does look like those with heme malignancies and more heme-like, like the lymphomas, have a more attenuated response in terms of the vaccine efficacy, but it's not 0. In fact, we're maybe 90% in solid tumors, it may be 60%. This is again based on early data. And my expectation is that you would see something, it's going to be a little less than you'd have with some of the solid tumors. With regard to the use of biologics, though, they found they got a very robust response. That was not, believe it or not, a modulator of this. Now, the other key thing that's really important, and we don't know much about this, is it a cell-mediated immunity or humoral-mediated immunity? Of course, it's both, right? We know that. And what we're doing with the vaccines is we've targeted the spike protein. These are, and really targeted for the humor response. I mean, that's where we're seeing the action, and the assays that are done to see whether the vaccines worked. You go back to what Pfizer did in Israel, you're looking at antibody titers here, right? So you see a big rise up. And then we also saw that with boosting, you got to rise up, and then it fell even faster. So the humoral pathway is not explaining what's going on with real protection. In fact, I think the vaccine companies probably shouldn't have made such a big deal about this, suggesting we might have sterilizing immunity. I think the T cell response is important. We're looking at that here. We're trying to look at T cell subsets and other attributes, because I think there'll be much less variability there in terms of the response, and probably the protection is the thing that's probably helping all of us prevent getting severe disease. But we don't know right now. Even the article cited these small studies and just some preliminary data. So we're gathering this data. We will be looking at that. So lymphoma patients will be enrolled in here. And honestly, I'll just share with you, I've said this in calls with the NCI, we're going to have to amend the sample size. We're going to have to redo this. First of all, I want the adult groups on. If we got an overwhelming number of adults in this study, up to 36 years of age, we need to make sure we can get enough kids there. So we might have to do that just on that basis. But I really would like to see this be a large cohort study, 1,000 plus. And I think we would have to justify that. And one of those justifications would be maybe looking 6 months out when we get some idea of what our accrual looks like, is that if we're trying to fill in some of those subsets, we want to make sure we have appropriate representation of lymphoma patients or folks in this age category. The argument would be that we probably need to rethink this. It is a cohort study. It's not a treatment trial. So the risks involved in this are not the same. I mean, remember, we do our sample size calculations to be efficient, but it's also a safety issue. And when you're doing epidemiologic studies like this one, the safety is not at the same level of consideration. So I'm hoping we'll be able to expand this out. It may become a resource issue. The NCI wanted us to do this very badly. I was given discretion to rebudget in our NCOR research base because it was really high priority. And the other problem about this is we got to do this quickly, because whatever we do in COVID, I've learned this. I mean, my COVID study ended in Davis in June. It's old news. It was in Politico 2 weeks ago. Nobody cares anymore because it's just so this is one of those things. None of us in oncology have ever dealt with anything that moved this fast. So I think it really is important to try to get up front high load accrual going, and then to be looking at this very carefully. The monitoring we'll be doing here, it's not safety monitoring, it's just monitoring to see what the numbers look like in terms of the population characteristics, because we want to make some inferences about the things you talked about, for example.
Thank you.
Sure.
Go ahead.
Hi, thank you for this. Really interesting. I serve as the advocate on the lymphoma committee. But my question is, and maybe some of the young adults that are here today can answer this, but what do you anticipate the compliance will be to get your blood drawn, at these frequent intervals when you may not be coming to clinic? You may or may not be still coming to clinic. And can they go to a local Quest lab or anywhere to get their draws done?
Yeah, that's a great question. And again, having been involved with the studies and looking at what happens with our AYAs, where do they go? Just in general, I've published that, so long ago. It is an issue. And I think we're going to see a pattern. I think we're going to see more complete data for the very young kids. And I think it's going to taper off as we get to the older ages. It's something that we have thought about. It's not an onerous schedule. It's every 6 months when you get out out past the kind of the immediate time. But we'll look at that. And again, we got this kind of funny relationship with the NCI having the contract with Quest, but your suggestion about being able to do this in their neighborhood, in their backyard, makes all the sense in the world. It's a very good suggestion because we want to make this incredibly easy for all the subjects. There should be no reason to skip this. It's a little bit of blood. It's not a big deal, but you're exactly right. So the more trouble it is if they have to come in to their treating center, they're done with their treatment, they're out of there. We may have some very bad follow-up. So thanks for the suggestion.
I have a question. Given that many unvaccinated AYAs have probably already had COVID, would you expect any background signal from previously infected people? It's humoral immune response should go up with a vaccination either way, right?
Well, that's where you're looking at the N protein and the S protein. Yeah, so you can discriminate natural infection from vaccine.
Yeah.
Yeah, definitely. And we're doing that. So we're doing those assessments. So it's fine if they were previously infected with COVID. It's not fine if they were vaccinated for COVID yet. So, but yeah, we'll have to sort that out. And again, that's another source of heterogeneity, what their history was prior to diagnosis with having a real infection.
And will you track, are you going to ask that as part of the study? Because that may impact their severe disease.
I think it's on the
the
intake form. Yeah, we have the demographic intake form that asks questions about their COVID history. So we are going to get that. I hope it'll be accurate, but yeah, it's important. Yeah.
Is there any racial disparities? I know there is in outcomes, and I didn't see that built into the study.
Well, actually, one of the tables I dropped was our accrual today. You'll look at race ethnicity, so we've got that. It looks like our incidents, like our patterns in terms of who's coming in. There are clearly differences. I mean, with the cancer outcomes, there are clear differences in kids as there are in adults, and it goes in ways that you wouldn't necessarily predict based on the adult outcomes. So we tend, the Children's Oncology Group, we're not perfect, but we tend to get a very representative chunk of the, because I would say, well, we only put about 60% of all young people on trials. That's still pretty good. But in terms of the folks that are interacting with us, it's still a very large chunk. Again, it changes through the AYA age period where they're not coming in.
The AYA is not near as representative.
No.
In fact, they're worse than adults.
Exactly. Yeah. And actually, one of the papers we published in Cancer 20 years ago was when you got to the 15 to 19-year-olds, we were only seeing 25%. This is based on a seer linkage study that we did back then. So it's definitely an issue. And again, this is going to be the issue about who's coming in to get their cancer care, where are they receiving their cancer care. This is, again, it's an NCORE. It's not an NCORE only study, but NCOREs we want to make sure our NCOREs are really engaged in this, that we're pushing very hard. That was what WARDA's desire was to do, and ours too, because again, that gets at a little bit more population representation, but we still have that issue of, they're just not coming, and their outcomes could be much worse. We don't know.
Yeah.
And unfortunately, none of the state cancer registries are adding COVID in to their to the information, which they couldn't even. We can't even figure out who's got COVID without cancer in California with our CalReady system. So it would be impossible to add that to the registries. Yeah.
I have a quick question. I'm the one of the AYA research advocates for this committee, and so the sites that you have enrolled, have you engaged within those sites the AYA clinics? Because I feel
Yes.
like,
Yeah.
okay,
Yeah. Actually, they're where we have them. Actually, you guys all know we're doing the AYA landscape survey, which we did. The other thing I did in my earlier days was I was one of the inaugural co-chairs of the CCDR steering committee for the NCI, and I know when we launched our first. It was the NCORE landscape survey back then. We're doing another one right now. There's a very strong pediatric component, but we actually have some demographics, if you will, on how our institutions are structured, including, those that, that we have SWOG sites that are part of cancer centers that have adult programs. But without question, we have a very strong set of AYA advocates, and many of our centers have AYA programs specifically. They're very engaged in this. We want to push it, but what we're seeing, at least and we have some planning meetings going on with the landscape survey data, is there's a lot of heterogeneity. You can call yourself a clinic, but in fact, you see a very small proportion of the total volume there are people in that age category. There's lots of issues like that, but there's no question. And the reason I'm here, and the reason from day one, we wanted to do this as an AYA study. That's why the age range went up there. It's going to be very interesting to look at that. And I am worried that their outcomes may be much worse than it is for some of the even older patients. Yeah, good.
Right.
Okay, thank you very
so
much. Yeah.
All right, Dr. Herrera says he's on the line now. So I'm going to see if I can.
Can you hear me okay?
Yeah, I don't know how to easily advance back, so I'm just going to go all the way back.
Sorry about the delay.
No problem. I'm glad you could make it on.
It would be too expected for me to complain about LA traffic. So.
All right.
Yeah, this is it. So I think it's just, I tried not to have too many slides so I could talk, although if they might call for me as my flight is boarding.
There you go.
Sorry about this. So thank you so much, Becky and the committee, for the opportunity to present today. I presented a few years back when we were starting this trial, SWOG-S1826, which is a randomized phase 3 study of AVD chemotherapy combined with either nivolumab or brentuximab vedotin in patients with newly diagnosed advanced stage classical Hodgkin lymphoma. So really trying to identify what's the best novel immunotherapy partner for kind of our standard AVD chemotherapy backbone that we use in adults, which is, I don't know if I probably can't advance the slide, so I probably have to ask for it to be advanced.
Yeah, I'll advance it for you. Ready?
And so, which leads to the kind of bulk of the presentation today, which is really thinking about and talking about how we did this and how we were able to collaborate with SWOG and kind of bridge this gap between pediatric and the adult management of Hodgkin lymphoma. So I'll be brief here because I think most of you know that about 20 to 25% of patients with advanced stage Hodgkin lymphoma will have relapsed refractory disease. And the standard in most of North America, I would say, has been ABVD-based therapy for advanced stage Hodgkin lymphoma, and the RATHL study showed that we could drop bleomycin in patients with PET2 negative, and that was non-inferior to just completing 6 cycles of ABVD. However, in the pediatric population, SWOG has had a different backbone for many years, ABV-PC, and they use response-adapted radiation therapy, which is associated with a similar kind of failure rate, but their regimen has led to the use of radiation therapy in a large proportion of advanced stage patients, more than half. So the Echelon-1 study evaluated BV combined with AVD and compared that in a randomized way to ABVD. And that showed, initially the primary analysis showed an improvement in modified progression-free survival, but now traditional PFS and even most recently, there was an overall survival benefit, which is the first time we've shown an overall survival benefit for many years in Hodgkin lymphoma in patients with stage 3 or 4 disease. But even then, there's still a significant proportion of patients who will develop relapse refractory disease. And so there remains room to improve, and BV-AVD is a bit more toxic. And that's just, there's benefit to seeing if we can improve on the regimen and with something maybe not quite as toxic. So the rationale for S1826 was to use nivolumab as combining that with AVD, and that was based on the fact that Hodgkin lymphoma is associated with overexpression of PD-L1, usually based on kind of ubiquitous chromosome 9p241 alteration in the Reed-Sternberg cells. So that encompasses the genes for PD-L1 and 2, and and leads to overexpression of the PD-1 ligands. And there's actually more 9p241 alteration in advanced stage Hodgkin lymphoma, and increased 9p241 or PD-L1 L2 alteration is associated with poor outcome with standard frontline therapies. So really established the rationale to see if we could use nivolumab to overcome that, the effects of the 9p241 alterations and their prognostic impact. And so we designed S1826. Next slide. And the objective of the study was to compare the PFS between the two regimens, Nivo-AVD versus BV-AVD in advanced stage Hodgkin lymphoma. And we also were looking at event-free survival, overall survival, complete response rate at the end of treatment, and safety and tolerability. Next slide. And so we did also include prospectively quality of life analyses to look at physician-reported treatment-related adverse events between arms, as well as patient-reported outcomes using selected PRO CTCAE items. And we also were looking at health-related quality of life, the PROMIS fatigue, FACT-GOG to look at neuropathy at various time points during and after therapy. Next slide. So this is just the schema for the study showing a 1-to-1 randomization for patients with newly diagnosed advanced stage disease, and they would get either Nivo-AVD for 6 cycles or rituximab-AVD for 6 cycles. We allowed for the use of radiation, but only in patients who had residual FDG-avid disease at the end of therapy, and they had to meet certain criteria. It really had to be that this was residual, low, a small burden of disease. They couldn't have they couldn't have progression. They couldn't have many sites of disease that would signify that the response really wasn't adequate. Next slide. So the study enrolled, patients according to the following eligibility criteria, and this is all pretty standard stuff. Newly diagnosed advanced stage disease. We allowed the enrollment of patients aged 12 or older, patients that have adequate performance status and organ function, and then we did allow HIV as long as it was controlled, and then kind of usual nivolumab related and brentuximab related restrictions. Next slide. So we've actually completed accrual about a year ahead of schedule, which is pretty incredible. These were the lead accruing sites, and you can see there's a whole range of sites across the country. The next slide actually shows, back one. The real incredible part of this study, which was the collaboration that we saw across the cooperative group. So I'll talk a little bit about it in a second, but we were able to engage COG from the very beginning of planning this study. And as you can see, that really led to incredibly robust engagement and participation. So SWOG was the leading enrolling group, but COG was right there with 32% of the enrollments. And actually, we also included the CCTG, the Canadian cooperative group, from the beginning of the study. And again, there were logistical reasons as to why they didn't come onto the study until very late, unfortunately, but they, within 1 year, were able to enroll about about 3% of the patients on the study. Next slide. So we have an awarded BIQ SFP to look at circulating tumor DNA assessment, and we are amending the study to include that. We also have planned imaging correlatives, and will be submitting grant applications to fund those that part of the study, looking at quantitative PET assessments like metabolic tumor volume and other kind of similar quantitative PET assessments. And there's potential to leverage both circulating tumor DNA and quantitative PET to metabolic tumor volume, for example, to develop prognostic and predictive models in advanced stage Hodgkin lymphoma that could help us design the next study. Next slide. So what are the potential impacts of this study? So this is the largest advanced stage Hodgkin lymphoma study in the history of the cooperative groups. The study really aims to define a new standard of care in advanced stage Hodgkin lymphoma, like most of our studies in the cooperative groups. the PROs and the other correlatives may allow the optimization of these, of kind of how we use these novel agents in the front line. And so what do I mean by that? So certainly it's a possibility that the study is not positive, but we may identify biomarkers that would suggest that one particular therapy or the other is better in a particular population. And I think a really tremendous contribution could be that if we see acceptable outcomes, this novel way of administering radiation just at the end of kind of reserving radiation for the highest risk patients who just have residual lymphoma at the end of therapy could really spare many young patients radiation that's associated with late effects. Next slide. So I think the exciting I'd say that the most exciting and maybe impactful part of the study was really this collaboration across adult and pediatric cooperative groups, in Hodgkin lymphoma. It's not something that we had done before, and we've treated Hodgkin lymphoma differently in peds and in adults for decades now. And so this was really the first time we did this. We had a planning meeting from the very beginning. And engaged Canada and engaged the pediatric groups to really build this together and get everybody's consensus and buy-in about what the best trial design was. And really from the beginning, it was really a goal to harmonize the treatment of Hodgkin lymphoma across adults, pediatrics, and the AYA spectrum. And this has led the groundwork for future collaboration. So the collaboration, went really well, and there was really tremendously positive, I would say, feelings about the collaboration amongst the leadership. There were some hiccups along the way in terms of how we rolled this out at centers that had, for example, participated in SWOG and one of the adult cooperative groups, and how that credit was assigned and things like that. But I think we were able to largely overcome those hiccups. And now there's a SWOG study that's approved by the Lymphoma Steering Committee for early stage Hodgkin lymphoma, the frontline treatment of early stage Hodgkin lymphoma. And that's a collaboration across SWOG and the adult groups. And then EA4211, which is the first salvage study for patients with primary refractory or relapsed Hodgkin lymphoma. That is also a collaboration across SWOG and the adult group. So basically this is the new standard, right? So I think that really, the collaboration, as you saw, I think really accelerated the research. We finished the study a year ahead of time. We can conduct these trials more quickly, answer questions more quickly, move on to the next question, and really hopefully advance the care of these patients much more rapidly when we're working together. And we've also observed that we can leverage the scientific strengths of each group. So our pediatric colleagues have been incredible collaborators in terms of guiding the PRO correlatives and survivorship studies. We have collaborators from Alliance who are leading the translational medicine aspects of the study, and they've been able to also be engaged on some of these other prospective studies that are in development because of the collaboration, and likewise with imaging and cost effectiveness. So it's been really nice to basically our goal was that every group would be able to lead a part of the study, whether it be the translational medicine, the PROs, the imaging studies. And I think that has also been a nice kind of standard to set or expectation to have. Next slide. So yeah, just to acknowledge, the last slide is just a conclusion and showing all of the groups that were involved. But I think this is, we don't know how the trial will ultimately, what the readout will be, and it's an event-driven trial. So we'll hopefully hear that in a couple of years. But no matter how you slice it, it's really been an impactful study in terms of changing how we design and conduct clinical trials in Hodgkin lymphoma in the cooperative groups. So thanks for your attention. I'm happy to answer any questions, although I will have to run in a couple minutes to catch my flight.
Awesome.
Sorry.
Thank you so much, Alex. It's such wonderful work, and kind of exactly what people were hoping for when the NCTN groups came together, bringing sort of SWOG closer into the mix. And you guys have really collaborated from the start and all the way through, and that's impacted the development of the future trials in Hodgkin. So it's just super exciting. We have time for one question if anyone has any. And if not, we'll let you go. Yes, Doc. Here's one.
Hi, Alex. It's Adam from University of Chicago, and I'm working on the AHA 2131. The question I have for you is, I think you're right, it's easily the new standard for lymphoma, but I think we're still struggling a lot in other disease groups. I guess what kind of from your experience working through this as one of the pioneers, what suggestions do you have? What do you think we can do to help with the other disease groups, specifically, leukemia and some of the other ones that likely, could benefit from it?
Thanks for the question, Adam. Yeah, I think there's always a bit of, I think change is hard, and there's always trepidation about figuring out how to make sure that everybody gets credit. Like, how do you balance all these things when you have a lot of I think that I wouldn't say they're competing interests. I think everybody has the same interest, which is to advance the science and advance the care of our patients. But there's always, we're all in academia. And so how do we make this all work functionally? And I think it requires a little bit of unselfishness, to be honest, and say, listen, like, as opposed to being the first author or the last author, whatever it is, of every single paper that comes from this, like, we're gonna, like SWOG, we're gonna lead the primary analysis, and maybe, and you just have to really be thoughtful and say, all right, COG, you guys can guide this part of the study. I mean, from the beginning, we tried to do that to make sure we had everybody's engagement, to get buy-in for really robust enrollment, and we were lucky in that, and that's what we saw, because I think the champions who were involved from each of the cooperative groups felt engaged. The COG was certainly engaged, and frankly, a lot of the feedback was that the regimen was simpler to use than the traditional regimen. So I think it's like just meeting from the beginning and building that trust and building that commonality and goals, and that we're all going to do this together. We're gonna do this study, and then we're gonna finish this one a year or two earlier than we would have otherwise, and then we're gonna work together, and you guys will lead the next one. I think that that kind of collaboration really is what we can aspire to, and it may be difficult to achieve, but I think when people see what we've done with Hodgkin, and then there will be a paper from the Alliance that leading, about S1826. There'll be a paper from COG about S1826. I think everybody seeing that can hopefully serve as a model.
All right. Thank you so much for this. It's inspirational, and have a nice flight.
Thank you. Take care. Appreciate it.
Take care. All right, next, Dr. Alan Rhodes from the Mayo Clinic, talking about current and upcoming AYA sarcoma trials. There you go.
Well, I gotta say, I'm very excited to not only present other people's trials, but with this finicky hair trigger, this is gonna be a trip. So thank you, everyone. Good afternoon. I am Wendy Allen Rhodes. I'm from Mayo Clinic. I am a pediatric oncologist, and I work within the sarcoma AYA committees at COG. And thank you for inviting me to talk about this. And we're gonna talk about some of the open trials and some of the ones that are approved but still in development. So when the trials are initiated, or the concepts are initiated through COG, this is sort of our process that, and I think this speaks to what Dr. Herrera was just talking about, is the concept is developed within the disease group, and there's opportunities for collaboration across the groups at these kind of starred time points at the very beginning when we're going through the concept development, and then our disease group has to approve the concept, and then it goes up to leadership in COG, to the scientific council. And then again, the next step of approval is at CTEP. And as I was designing a trial and going through this approval process, by the time I got to CTEP, Dr. Seibel was really adamant that had you talked to your adult collaborators, and had you gotten buy-in, and had they been a part of the development of your trial. And so I think we're seeing more and more that the development and the involvement across the groups is getting pushed further and further back from the very beginning. And I think some groups are doing it very well, like Hodgkin's lymphoma, and then there's some people that it's kind of happening later, but I think that paradigm is shifting. So I divided this up by sort of discipline into bone and soft tissue. So we do have currently 2 open and enrolling osteosarcoma trials and 2 approved osteosarcoma concepts, and 1 is approved for Ewing sarcoma. So the first trial that is open and enrolling is AOST 2031, which is led by John Doskey, who's a surgical oncologist. And this is a randomized phase 3 trial comparing open versus thoracoscopic management of oligometastatic pulmonary disease in patients with osteosarcoma. And the primary objective is to determine if open surgical resection is superior to a thoracoscopic approach for thoracic event-free survival in patients with resectable oligometastatic disease. And oligometastatic disease is defined as 4 nodules or less per lung. Secondary aims are to determine the patient event-free and overall survival in this cohort, and then to determine if the thoracoscopic is superior, if thoracoscopic approach is superior to open surgical resection for postoperative pain. So this is the first osteosarcoma trial where we've been successful in embedding patient-reported outcomes into the trial, which has been very exciting. This trial is open for patients up to 50 years of age. They have to have metastatic lung-only disease, so no bony disease. They can be either newly diagnosed or at first relapse. That's the definition of the oligometastatic disease. And they have to have no prior thoracic surgery for metastatic disease and no disease progression. This is actually a pretty short trial. It includes enrollment and randomization with central review of the chest CT and then surgery within 28 days of that chest CT. The post-op observations are all completed within 3 months, and those are those PROs, which are done from the coordinating center through a red cap. So it's a little bit of a burden on the site up front for the CT and the central enrollment, but then after that, it gets easier. Some of the barriers to enrollment have been this very tight turnaround window of 72 hours from CT to enrollment. And so the word on the street is that in the next amendment that will be loosened up a little bit. And then one of the other barriers to enrollment has been the no prior thoracic surgery. And so I think they're also going to loosen that up to say that if you had a biopsy to prove metastatic disease at relapse or at first diagnosis, that those would still be able to be included later on down the road. The trial is powered or is estimated to enroll 250 patients. It's a long trial, so we went from very fast with our COVID trials that Dr. Pollack talked about, and then AHOD finishing early, and now we're back into the 5-year trials for sarcoma, so it's a little bit humbling. But there's 6 strata on this regimen, broken into newly diagnosed, first relapse with the intention to give chemotherapy afterwards, and then first relapse with no chemotherapy. Each of those is broken into poor and average risk, and poor risk is defined as 2 or more metastases, poor necrosis in the primary tumor, less than 18 months from the end of therapy to the first relapse, or bilateral lung metastasis. And it was powered to detect a 15% margin of superiority with the open procedure compared to thoracoscopic. So that is open and enrolling, and there is a big push. If you listen to any of the COG surgeons, there was all sorts of social media pushes, and there was some tattoos at COG temporary to push this trial because we really do want to answer this question. And we're going to try to hopefully make it a little bit easier on the sites for enrollment. The next open trial is AOST 2121. I have to look at the numbers because these are not my trials, so I get a little bit turned around. And this is an industry-sponsored trial looking at OST 31164 as maintenance therapy for patients following resection of recurrent pulmonary disease. The primary objective of this trial is to determine if administration in a maintenance type of fashion of this biologic agent increases disease control, and also to assess the tolerability and safety of this biologic. The eligibility is 12 to 39 years, so it fits in that AYA range very nicely. You're probably wondering, what is this agent? It is a live attenuated bacterial vector with a listeria bacterial vector that has additional plasmid and epitopes. I'm not going to get into what those are because above my scope of understanding of science, but those are meant to mimic acute infection that leads to reduction in the immunoregulatory T cells that leads to a more immunoreactive tumor microenvironment to target those micrometastases. This is an every 3-week injection for up to 48 weeks, as long as there's no progression or undue toxicity. And right now, this is a fully funded industry trial, and it is limited to 19 sites, so it's not super available across the country. And per the latest memo, there is not current interest to expand to additional sites from the sponsor right now. That may change in the future. But it is out there as an option for your patients if they're looking for something and want to travel if you don't have it near you. So the next study that we're going to talk about is AOST 2032, and this has been a very long process and in the making. I think I remember this at one of my very first COG meetings that I went to, that it had a different number then. But this is going to be our upfront phase 3 all-comer trial for osteosarcoma. And essentially it's looking at the addition of cabozantinib to MAP chemotherapy for patients with newly diagnosed osteosarcoma. There's a dose finding and a feasibility phase in part A, and then part B is the efficacy phase. So this is going to be eligible for patients up to 40 years with newly diagnosed localized or metastatic disease. And as you can see, if you look at the target accrual, it's a pretty hefty trial, and it's going to run for quite a while, but we hope that it will answer a definitive question. You can't see this, so I'm going to skip that one, but I guess I can show you. Oops, now I've got the trigger here. So on your left hand, anatomic right, is standard risk, and standard risk is defined as localized or resectable tumors, non-pelvic. And then once they're into standard or high risk, then they're randomized to arm A and arm B. So you're going to have standard risk and then high risk and then randomization within those once we get to the efficacy phase. So this is a little bit easier to read. This is the localized resectable non-pelvic tumors. So arm A is just going to be MAP as you know it, old, tried and true, still wishing for something better. And then arm B is going to be the addition of cabozantinib. Is this a pointer? Yeah, there we go. Cabozantinib here on the bottom. So this would be just the MAP, and then this would be the cabozantinib added in. There's a break in here during local control for concerns of wound healing issues. And then there is a 6-month cabozantinib maintenance on the end. If you look at the patients with metastatic, unresectable, or pelvic tumors, they have a little bit of an additional break in there for the resection of the metastatic disease. And so cabozantinib won't start until at least 2 weeks after the last thoracotomy to again account for those wound healing issues. So this is A, and then, or I'm sorry, C, and then D. So moving on to AOST 2221. This is actually championed by Jason Glover and Laura Davis, who I'm sure you guys know. This is a phase 2 study looking at adjuvant regorafenib in completely resected and recurrent osteosarcoma. This protocol concept has been approved with stipulations by the Scientific Council. So when we have stipulations, generally we can get them through, but sometimes it does kind of stop at that Scientific Council phase. So as you know, regorafenib is an oral multi-tyrosine kinase inhibitor that's shown efficacy in recurrent osteosarcoma with measurable disease, but we haven't ever tested it as an agent for maintenance chemotherapy. And so that's what this trial is looking at. And then moving on to Ewing sarcoma, there's one protocol that's received approval with stipulations from Scientific Council and has the number 2231. It's also gone through several iterations, and it's led by Dr. Bhuvanassetti from Nationwide. And essentially, this is also looking at regorafenib, but adding regorafenib upfront and in maintenance to patients with newly diagnosed metastatic Ewing's or Ewing-like sarcoma. So if you think of it in an easy way, it's VDC-IE, which we know for localized and metastatic Ewing's, versus VDC-VIR plus maintenance. Uh-oh. We have a ghost slide. It's okay. So on AEWS 2231, one thing I wanted to point out that for particular interest to this audience is on our COG sites, we're requiring enrollment onto our registry, but for non-COG sites, we're accepting central review of molecular pathology report for Fusion partners. So that's not going to be an issue if you're not a COG site. Yep, this is a fun pointer. So patients will enroll and start induction chemotherapy with a cycle of VDC, and then they'll have that central testing or central review of their testing from that site. If you have the pathognomonic Ewing sarcoma, so the EWS or the EWSR1 ETS fusions, or EWS fly, those things, they'll get randomized to A or B. If you are an eligible round cell sarcoma that is not Ewing sarcoma, previously known as Ewing-like sarcoma, now known as B-core mutated sarcomas, CIC rearranged sarcomas, or I'm forgetting one. There's one other one there that used to be in the Ewing-like. Those will get non-randomly assigned to regimen A, and that'll be the first time that we're testing these Ewing's-like or the new classifications that they have in a prospective fashion to understand how they're doing on Ewing's therapy. If you don't have any of the criteria for those eligible ones, then you're coming off protocol therapy. So this, again, is just showing the regimen here. Regimen A is the VDC-IE, as you probably know it, with 14 cycles, not 17. If you're into the Ewing's literature, that was something that was looked at on 1031, but we're going back to the 14 cycles for the metastatic patients because they didn't have any improvement in outcomes in dose compression or intensification. And the regimen B will be VDC-BIR. This is an easier way to look at it. This is what we all recognize as VDC-IE for Ewing's. And then down here is the experimental arm where you get VDC. Week 3, you get VI, which is the vincristine irinotecan, and then starting on, I think it's day 8 through, it's over here, day 8 through 21, you get the regorafenib. And then the maintenance is just continuous regorafenib. So that rounds out the bone tumor protocols. So that's probably, since I've been involved in the sarcoma world, that's as many open and in development bone tumor protocols that we've had at any one time. So hopefully they will all get through scientific council and CTEP review. So moving on to soft tissue sarcoma, we have, which is everyone's favorite, soft tissue sarcomas. We have 2 upfront rhabdomyosarcoma trials, one for low risk and one for high risk. We have 1 protocol that's open for relapse recurrent refractory desmoplastic small round cell tumor. And we have a group that did some AYA rhabdomyosarcoma outcomes retrospective review and study that'll be presented at CTOS. So I don't have the information for that, but I'll just give you a heads up to look out for that. So ARST 2032, I'm going to blow through pretty fast because I think it's a pretty narrow age range for people that that will be involved in this room. But this is looking to redefine low-risk rhabdomyosarcoma using clinically defined risk factors along with molecular biomarkers. And the intent is to de-intensify therapy for patients with very low-risk rhabdomyosarcoma without poor molecular features, and they'll get VA for 24 weeks, and then intensify therapy for patients that have what we think are poor molecular features with MyoD1 mutations or TP53 mutations, and they'll get VAC for 42 weeks. Oh my goodness. So this is the narrow age range I'm talking about. So if you're at an adult-only site, you're just going to get those 18 to 21-year-old. They're the lower risk patients, the stage 1 clinical group 1, 2. If you're clinical group 3, it's orbit only, and then stage 2 clinical groups 1 and 2. So this probably means the older adolescent young adult male with paratesticular rhabdo, completely resected, no lymph node involvement. That's the kind of patient that I can envision at an adult-only site. And then if you're at a mixed site, you'll start to get into that younger AYA population. Again, we're kind of slow in the soft tissue sarcoma world. For 194 patients, it's going to take us about 5.5 years to get those patients. This is the schema for the trial. So they'll enroll if they have very low risk disease, they're going to go on this VA for 24 weeks all the way down. If they're low risk, they're going to get VAC VA for 24 weeks. Once the molecular studies are back, if they have P53 or MyoD1, they're going to get called back and reconsented and go to regimen M. Oh my goodness. So this is what the VA looks like. Things I think about for AYAs that Laura Davis has taught me is all of this vincristine is how will people tolerate that in the older AYA range. So it is a little bit vinca alkaloid heavy, just so you know. Again, this is also very vinca alkaloid heavy for the VAC VA. And, unlike some of our other protocols, when we're really de-intensifying therapy, it makes me pretty nervous to start dropping and dose reducing if we're already dose reducing or removing therapy. So just something to think about. And then regimen M should look familiar to some of you. This looks just like the ARST 0531 regimen A backbone. And so it's VAC for 42 weeks at an intermediate cyclophosphamide dose of 1.2 grams per meter squared. And again, up front, they're very vinca alkaloid heavy until about week 13 when breaks start to happen. So just something to think about for your patients in that AYA range. ARST 2031 is our high-risk study, and this is basically, you can think of it as vinca alkaloid intensification. This is pretty heavy in the AYA range just based on the pathology and the epidemiology of high-risk rhabdomyosarcoma. So this is eligible for, or this is open to patients up to 50 years of age at the time of enrollment. Any patient with stage 4, group 4 disease of any subtype except adult type pleomorphic based on institutional histopathologic classification is eligible. We are doing fusion testing, so you're enrolled based on spindle sclerosing, which would be sort of fall in that embryonal or alveolar. Anything except adult pleomorphic can go in, and then the fusion testing is required to be uploaded by week 4, but can be done at the institution and uploaded a report. It doesn't have to be done centrally. This is how it looks. We have completed our safety phase. We had a 8-patient safety phase, and in that safety phase, we had a third of our patients were in the AYA range, and 3 of them were in their 20s. So we had a good enrollment, and that was all on the vinorelbine arm, which I'll show you. And now we're at the point where we're stratifying by histology and randomizing to either regimen A with VAC or regimen B with vinorelbine, dactinomycin, cyclophosphamide. Y'all, this is crazy. So regimen A, you should recognize that this looks very much like regimen M because it's the exact same regimen. So it's a vinca alkaloid heavy for 12, 13 weeks, then you start to get breaks. So it's just VAC, VVV, like we all know. There's primary site radiation at week 13, metastatic site radiation at the end. And then regimen B is substituting vinorelbine in for vincristine. So you get 2 weeks of vinorelbine and then 1 of vincristine. If you look across, you're getting vinca alkaloid for 42 weeks. That might look a little bit scary, but the safety profile of vinorelbine is more myelotoxic and less neurotoxic. And in our safety phase, we didn't have any grade 3, 4 neuropathy, including in our patients in the AYA range. So we felt it was pretty tolerable from a neurotoxic perspective. And if you compare the doses of vincristine, there's half of the vincristine in regimen B compared to regimen A because of the vinorelbine being added in. Maintenance looks the same as what was seen on ARST 1431 or the RMS EPSSG 2005 study, which is 3 weeks of vinorelbine and continuous oral cyclophosphamide, and that will go for 6 months. And both regimen A and regimen B get maintenance if they haven't progressed by the time they get to maintenance. It is currently open and enrolling. We are powered to detect a hazard ratio of 0.61 with a type 1 error of 0.1. So it's a little bit generous. We had to do that. We had to give some concessions on the statistics. The duration for this is about 30 months with about 4 enrollments per month, and we are hitting that accrual and with a follow-up of 2 years. So again, still a 5-year trial by the time we get mature data. We have one trial that is open and enrolling for desmoplastic small round cell tumor as part of a Lilly master protocol. So this is a completely industry funded trial that's meant to serve as a platform to look at novel agents in the Eli Lilly pipeline, alone or in combination, for patients in the pediatric and AYA range. And their aim is to have this master protocol and then kind of slide things in and out to test more rapidly and efficiently. There were 2 protocols that were open on this. One was for synovial sarcoma and the other one's for DSRCT. There is no efficacy signal in the synovial sarcoma, so that one's already closed. But 2021A is open, and it's looking at ramucirumab, which is a VEGF inhibitor, or monoclonal antibody rather, in combination with cyclophosphamide, oral cyclophosphamide, and vinorelbine, compared to cyclophosphamide and vinorelbine alone in patients, pediatric patients and young adults with relapsed refractory recurrent desmoplastic small round cell tumor. This has been very slow to accrue. I think we've only had 1 accrual on this so far. And I think some of the barriers to that is it just being an industry-funded study and some of the hiccups that come with opening that. It is open for patients up to age 29, and they have to have received at least 1 prior line of therapy and have measurable disease that's not eligible for surgical resection. And then lastly, just really quickly, Doug Harrison is going to present this data at the CTOS meeting in Vancouver in November of this year. So that data, I guess, is, embargoed for dissemination until after the meeting. But what they did was they looked at the AYA age range on the impact of the AYA age range on overall and event-free survival compared to children under 15 years of age for patients that were enrolled on trials from 1997 through 2016. So it was all of the low risk, intermediate, and high risk trials from North America, and looked at the outcomes and did a multivariable analysis to see if we could pull anything out of that. So that data will come out in CTOS and should be already out in abstract form. And then after the presentation, that will go on to publication. So be looking out for that. There's not a lot of surprises, unfortunately. Oops. So thank you everyone for your attention and me going through other people's studies. It's a little bit hard to give other people's updates, but I appreciate the opportunity. And if anybody ever wants to be involved in the development from the sarcoma side, from the SWOG studies that are initiated or want to collaborate, please just reach out. Everybody seems to be really nice in our committee. So just let us know if you guys are interested. And if you have any questions about any of the trials, all of the PIs are very nice. So reach out to them. Thank you.
Thank you, Dr. Ellen Rhodes. Any questions for her? I've got one.
Oh, yes.
So the age criteria for the different studies are kind of all over the map. And so would you talk about the priorities in your committee and why you think there are differences?
Yeah,
If that's clear.
so for the low risk trial, we picked 21 just because once you get beyond that age, number one, you don't see a lot of patients with low risk disease that are older, and de-intensifying for an older population felt unsafe because of the outcome disparities that we see. So that one was kind of loosey-goosey and picked that way. Originally, when we wrote ARST 2031, I had picked 30 as my upper age limit, and Nita Seibel was like, why did you pick 30? And I was like, I don't know, everybody else picked 30 before me. I thought that's what we do. And she was like, why don't you ask the adult groups what they want to do? And Laura Davis said, well, if your neurotoxicity is going to be less, let's try 50. So sometimes there's not a lot of science, but it's that kind of expert opinion consensus of, yeah, we really struggle with VAC in the AOA and the adult population, and we're going to struggle with regimen A, but maybe regimen B will be better. So there has been sort of this moving target throughout, and I can't always explain it, but those are two examples that I know.
Nice. And as in Hodgkin's, people are looking to the SARC collaboration and learning from that as you go along. Can you say a little bit about that group and what's going on there?
Yeah, so there's a AYA NCTN sarcoma working group. We used to meet monthly. We're now meeting more like bi-monthly or quarterly, depending on how many protocols we have to review. But there's representation from COG, ECOG, Alliance, SWOG. Anybody is welcome to join, by the way. And we just go over protocols that we're working on and get feedback from each other. So this is where I presented ARST 2031 and got the initial feedback from Laura Davis, and then took that back to CTEP. And CTEP really loves those working groups and is a big proponent of those. So I don't know how many exist in other disease committees, but it's been really successful for the sarcoma group. And it sort of fed off the success of Aaron Weiss's pazopanib trial and the co-development of that. So I kind of just slid in on the back tails. That's not my group. I don't claim fame to that, but I have enjoyed the benefits of it.
Nice. Great. Thank you so much. Get this. And next, we have Dr. Adam Duvall from the University of Chicago, who is going to be talking about gaps in AYA oncology fellowship education.
Yeah, thanks for having me, Becky. And I think I was here, how many years ago? So I did training in both adult and pediatric hemonc. I did fellowship in both. And so focused on adolescent and young adult oncology. See mostly lymphoma and leukemia as primary treatment, but my research is mostly in psychosocial care, health-related quality of life, and patient-reported outcomes. And so with a larger group that I will acknowledge at the end, this is, I would say I helped with some of it, but I don't think I was the leader of this by any means. There is, we basically are trying to, get a consensus for what education is going on in fellowship programs for AYAs with cancer. For fellows who take care of AYAs with cancer. So we all know that AYAs with cancer are very unique. They require kind of their own teaching to learn about. We really need to know about the different factors that impact their outcomes and both, actually morbidity or mortality, but also morbidity and quality of life. That includes all the different biologic factors, which we're really, failing to understand a lot about, but gaining more knowledge every year, I think. But also the financial, psychosocial, and almost most importantly, the developmental that they experience. So, there's lots of people, and most of the people in the group have written on this. Brandon Hayes Latin was one of my old mentors. He kind of really was one of the first people with a couple other leaders in this field that wrote about, what a training program for people who take care of AYAs with cancer should include, at least, and that was back in 2010. But it's been 12 years now, and we were kind of with the goal of trying to see where we're at after that publication in 2010, and a lot of the work from different organizations and kind of the more popularity of AYA oncology. What is actually going on in our fellowship programs. But it's because we don't know. We don't know exactly what is being taught in our programs and where the gaps are and what people want to be taught. Oh man, you're right. This is this is something. And I'm going to see. Okay, good. And so, basically what we're doing, and is led by Scott Mordler. And if you were at SWOG, he presented on a similar data, I believe, as well. So it's looking at basically where we're at. And so talking with our fellowship programs, both adult and pediatric, where are we at? Where are the knowledge gaps for taking care of AYAs with cancer? And what are we doing well, maybe? Let's be positive there. And so identifying that there's limited data, and that's really where we need to start. Before we can hopefully make things better, we should know, where we actually are. And so we want to look at the prevalence of AYA-specific subjects in training programs. And there's lots of different things that are encompassed in that. And so you'll see, and I apologize in advance, all the graphs might be a little bit difficult to read, but I just really want you to see the themes of it anyways. And that's because there's just so much that's so varied about taking care of AYAs with cancer that to ask a simple kind of topics within that, I think is going to do it injustice. So we narrowed it down a little bit, but you can see there's still a lot of different things that I think it encompasses. So what we did is a web-based survey that was through Rutgers and Scott. He was able to use it through RedCat basically, and a lot of it is looking at asking both program directors and fellows, and then really anybody who would be willing to answer us, because you know how easy it is to get people to answer anonymous surveys in a lot of ways. So we emailed invitations to everybody, and then Brandon and I and Scott and everybody emailed the program directors one by one with personalized requests to please just tell us what you're teaching your fellows about AYAs with cancer. And you can see we were largely not very successful, but we got enough to, I think, at least give us a background to start from. So you can see what we got back were pediatric program directors were great, and so were pediatric fellows. Our adult colleagues, which I am one of them, so I can't complain too much. They didn't quite answer as much, and so we only got about 27% of the programs that we were looking for to respond. And again, this is after multiple emails that were personalized to the program directors and everything else. So we kind of at that point said, okay, this is about as good as we're going to get. Not surprisingly, most didn't have any formal curriculum. And if you look at just the program directors, again, most don't have a curriculum. And the ones that did were all pediatric and one that actually had an advanced AYA fellowship. I think we all know which one that was. Oh no, we tried to get this fixed, but for some reason it's not. So this is supposed to look like this, where you can see basically based on colors where the gaps are that we're missing. And so what I can tell you is basically that I'll go through here. You can't, and again, I apologize. We literally just looked at it and then tried to redo it again, but I guess it didn't work. So if you look at everybody who responded, and you look at the amount that people are getting, there's basically not very much. The biggest one that the biggest amount that people get is in oncofertility. So there is, and there's been a lot of push. There's a lot of organizations for it. So what you can see is that what we took it as is that it's actually effective at least. So oncofertility was much more prevalent to be in education than the other subject matters. And then you look at basically pediatric versus adult. This gets back, and you can see there's actually, even with the small numbers, were a good amount of statistically significant differences between them. But really what it amounts to is kind of my personal experience being trained by both is that pediatric folks are much more interested in AYA oncology, or they at least have more time to devote to it. So there's a lot more education in pediatric programs than there is in adults. That's what all these differences are, is that there's just more of it in really any of these subject matters. But again, if you look at the numbers, adult providers are seeing more AYAs than our pediatric counterparts. And so that is lovely, but it's not really helping our AYAs and by educating the providers who are actually probably going to see them in the community. If you look at program directors, the only, versus fellows thought there was a lot less education going into AYA oncology than the program directors did. And that was different enough in oncofertility, but the other ones, it was you could see that there was a difference in perception at least. And then if you look at people who had, this is, pretty self-explanatory, but basically if you had an AYA specific program, which again, were just very few, so that's why there's not any statistically significant differences. But if you had an AYA program, you are much more likely to actually have formal curriculum about AYA specific issues than if you didn't. And, basically, pediatric providers are more comfortable talking with AYA specific topics than adult providers in a general theme. Okay, so now you can actually see it, but the colors are really what is important. And you can see this kind of goes throughout. And interestingly, there are differences in what our pediatric versus adult colleagues kind of feel about what's important. So our pediatric folks think really everything is kind of more important than what our adult colleagues felt. That probably is reflective of what they're actually doing as well. So that shows us that we're probably not doing a good enough job, talking about this and reaching at least the general adult community, because this is really an unselected fellowship program. So the people who are leading these programs, we're obviously, whatever we're doing, we're not doing a good enough job in reaching them, or at least, emphasizing the importance of AYA-specific education for their trainees. When you look at the program director versus fellow, though, they're all about the same. And this is what we're really looking at. We're just looking for general trends. There wasn't like the fellows thought something was much more important than program directors. They were kind of all the same. So if we look at, where we can maybe make an impact, it could be potentially in kind of trying to work or trying to educate or whatever, or convince, I guess you could say, our adult colleagues, or the adult leaders of fellowship and training programs, that this is something that's very important, that this is something that we should include, or at least make it maybe possibly easier for them to do so, which we'll talk about later a little bit. And so again, if you look at people who have formal AYA curriculum, they all think everything is much more important for the most part than people who don't. Obviously, they designed an AYA curriculum. It's kind of stating the obvious a little bit. So when we talk about, putting it all together, and what this looks at is basically, all the different subjects and what the importance is versus the current amount. And of course, you can't see the current amount. It's not projecting, so it's not projecting here either. But again, my apology, I don't know why it's not working. But so basically, everybody thought that it was more important than what they're getting. Is what it amounts to. Even for all of those different things that we talked about, the differences between pediatric, the differences between adult, people still think it's more important than what they're getting. So we do have an opportunity, I think, to actually impact this if we can maybe, design resources or implement something that makes it a little bit easier to access. So, basically it's lacking, but it's more absent in adult training programs. They believe that the AYA topics are important, but even though they're infrequently taught. And the most important topics they saw were oncofertility, survivorship, and then communication. Psychosocial issues were kind of right below that. Biology was more important in the adult side. But really, we need to kind of take this further. We're actually writing this up for publication right now with graphs that you can read, and are kind of planning our next steps with this group that we're doing. And those next steps are really, we talked about, is is the next step to kind of get more of this information? Should we be also, talking to the people who are actually seeing these people on a one-to-one basis, our nurses, our APPs, our social workers, getting all of the all of the ancillary staff that are likely more important than the MDs anyways, and seeing what their opinion is, and if they're, from their institutions or from their education and their training, what education they're getting. And then, talking about actually the quality of their curriculum and things like that, that would be another point that I think we're leaving a little bit in the wayside. But what we're really doing is actually now trying to develop curriculum. So there will be potentially anybody in this room could be contacted where we're developing curriculum that will focus on those specific topics, attempt to, utilize relationships and leaders in the field to design, basically talks that can be easily disseminated to these different programs in each subject matter. And then we're going to, be asking people to kind of review them and see and comment on them so that we can get them to be the best of their ability. And that's really what we're working on now. And if you want to join the group, please reach out to myself or Scott Mulder. And, we're really in the infant phase of it. So we're talking about, just pre-recorded videos or slide decks or a lot of different ideas that we're working on that could potentially remove a barrier to be able to get these topics in our fellowship programs. Interestingly, we figured out about, 6 months, almost a year in, that non-malignant hematology through ASH, we're working on the same thing. So specifically for, hemophilia, sickle cell, all of those topics, which are obviously equally, if not as more important in the AYA space. And so we're collaborating with them about the same thing. So we're trying to now develop, curriculum that will be mirrored similarly in the non-malignant heme and also in our oncology programs. One being worked through mainly in ASH, and then one kind of being separate. We're kind of housed within the COG, but we really don't have a home at this point. We're just kind of a group of people who are working on it, honestly. So at that point, I'll pause. Okay, this is basically what I just said. So we're doing a virtual AYA seminar series, and then basically a toolkit with, again, lectures, pearls, and articles, kind of just what I said before. So these are all the important people. Scott has really been, whenever we get off track and get distracted, Scott is really the one that brings us back and is really a leader in this and doing incredible work. And then everybody else who's involved has also been wonderful to work with. So I'll take any questions. And again, I invite collaborators because we're really, this is really what I think we're using. We're using our baseline data that obviously is limited, but it's really just trying to illuminate what we already know. I think we all know anecdotally where our limitations are. And so we're just trying to use that to really focus on making those decreasing those barriers and hopefully giving resources for our program directors.
So thank you. I think this is great. I guess one of the challenges, I'm just asking you, is the age range of AYA is so huge, right? And the needs of those patients, both clinically, emotionally, everything that's going on in their life developmentally is so different if you're, you've reached puberty at the age of 12, or if you're 30 years old and you're on to your career path, might be thinking about having a family or whatever. So there are such huge differences in terms of the needs of this huge cohort that we lump into one. How do you address that in the training to make it usable and valid for the different experiences that these clinicians have and the different cohorts that they're treating? It really varies.
Yeah, and I think, I like to say, and I think they've used it in the older population when talking about intensive therapy and things like that, but age really is just a number too. Right now, I'm actually on our inpatient service. I'm attending our leukemia service right now, and we have a 27-year-old who you would say is very equivalent to a lot of the 15-year-olds that you see. And then we have 18-year-olds who see us in clinic who have multiple kids already in our job, working full-time, and wife and kids and all that kind of stuff. So yes, the age range is wide, but even within that age range, the number necessarily doesn't even mean anything. So what we're trying to approach it as, and it's really just thinking about hard skills and soft skills. So, what are the hard skills that someone needs to be able to approach somebody in this age range with knowing that wide wide differential, and just at least some of the, and be able to meet them where they're at, wherever that may be, through that wide age range. And with communication and other things that, there's just some kind of basic skills that would be good for anybody, whether or not they're AYAs or not. But then, some of the other things would be looking at, just some of the background knowledge to know that, the fact that not everybody knows that, right? So just teaching people that, is a win in and of itself. So just trying to get some basics, some building blocks to be able to then, our patients, I think, educate us over, decades of work, basically. So I did as much training as anybody, but I wasn't ready to, be taking care of AYAs by myself, just starting off anyways. So it's really just trying to get those basics so that when they go out and start practicing, that they're able to then learn from their patients and be able to, grow in their career. So it's really low goals of just trying to get some basic stuff that hopefully, if that's something they either are interested in, it sparks them to do more, or if they're not, it at least gives them a baseline that when they encounter it in real life, they're not surprised, or they handle it in a way that's going to be a positive experience for them and their patient.
Other questions for Dr. Duval? Thank you so much.
Yeah, thanks.
Really interesting. And to close the session, I wanted to just touch on a project that Jonathan Summers and I, who's here in the audience, and others did a couple of years ago. We had a group of AYA patients convened in Tacoma, Washington. And during this PCORI project, they met monthly for a 2-year period. And there were not only patients, but also a spouse, sibling, caregiver, and some parents. And they talked about their unmet needs during cancer care. And so this is a very different and patient-focused way to come at this exact same question, really, is what do patients want their cancer care to look like, and what do they want their providers to know? And so we made a video based on those discussions that touches on those subjects, and I think interestingly reaches a lot of the same conclusions that you find when you ask program directors and fellows what they want to learn about. So we wanted to play that for you. This is a picture of the filming of the video. Interestingly, these PCORI grants are iterative projects, and so rather than being hypothesis-driven projects that get funded and then you do specifically that thing, we were funded to convene this group and see what they came up with. And one of the things they wanted to do was to make this video. Jonathan.
I just want to say one quick thing. Hey, I'm Jonathan. I made this book. So they made a video first, and I watched it, and then with my producing partner Eric, and thought of all the things I wish we could tell a doctor, all the things, and reframing it. And then we came up, as you'll see, this video is called AYA University 102. But the whole idea is to create a series. This is just one class, and AYA is not really a university. It's just something I thought of at 3 a.m. when I couldn't sleep. But I hope you enjoy it.
All right, so we'll show this. It's about 5 minutes long. That'll take us to around 5:30, and Jonathan and I will be available for questions after. So thanks so much for attending this meeting. Let's see. I'll just go through these and get to this.
Everyone, this is AYA 102, Patients Teaching Professionals. Please take a course syllabus and pass the rest to the person behind you. Patients Teaching Professionals is designed to help all clinicians improve the outcomes for these patients. We have visiting professors that are truly experts.
AYA, that crazy acronym, is adolescents and young adults
who have been diagnosed with cancer between the ages of 15 and 39.
There may be only a few AYAs in your clinic,
but there are more than 70,000 of us that are diagnosed every single year in the US.
According to the National Cancer Institute, our rates of survival have not increased since the 1970s.
It's 50 years. That is a huge problem. And it has been improving for other ages.
We also have by far the lowest rates of participation in clinical trials.
Many clinical trials aren't even designed for AYAs.
If all of this doesn't bother you, it should.
It really should.
AYAs are unique because we're in the prime of our lives.
A lot of us are in the middle of huge transitionings and big decisions.
My freshman year of high school.
2 weeks after my 21st birthday.
I was diagnosed at 30 when I gave birth to my first kid.
And in a weird way, it takes away your innocence. Like, it takes away your ability to think about the future. Like, the future is a language I don't speak.
I went to the adult world, and everybody had gray hair and was basically dying. They took me to the children's hospital, and I had to share a play kitchen with a 2-year-old, and I just knew that I didn't belong anywhere.
Oh, let's talk about how to tell somebody that they have cancer.
Ooh.
So how to tell me that I have cancer? We feel like this is really important to bring up with all providers out there.
So we want to share with you how to do it well.
Not a single one of us had a really positive experience being told that we have cancer. And it wasn't even that we were being told we had cancer. It was how they told us we have cancer.
Nobody told me. Everybody thought somebody else did it.
Some of us were told on the phone. We don't want to be told on the phone.
Set a time and a place to meet. Let me invite my favorite people to be with me.
Those people are going to make me feel more comfortable in the situation itself.
Before you go in, find your center. Take a minute to yourself. This could have happened to you, but it happened to me. Take a deep breath, walk in the door, and please sit down with me.
It's amazing how many of us were told that we have cancer by a doctor who was standing up and walking out of a room.
They call me Mr. Harrison. Felt like my dad. Call me by my name. Touch me on my arm or shake my hand or something. Make me feel more comfortable.
When you talk to me, please remember, I may know nothing about healthcare.
Show me that you're the expert.
And if you're not the expert, how you're going to find an expert
Exactly.
who knows that. Include the numbers and percentages that are in your head.
I will Google my own diagnosis within 5 seconds after you leave the room.
Be kind, give us time to take in what you're saying and ask questions.
Tell me what you do for your family member, and then we'll make a plan together.
And if you don't know something, dear God, say, like, here's what I know and here's the stuff I don't know.
Because it'll build a shared relationship between us.
Offer me a clinical trial.
Or tell us why you're not offering one.
No matter what it is, let me know that I always have a choice and that I have choices to make.
Because I am choosing if I want to pursue treatment or not, and that's a choice. And when you give me a voice and let me make decisions, I feel like I have more control over my situation.
Be strong enough to look me in the eye.
Imagine your brother, your sister, someone you love in our place.
Don't be afraid that I'm gonna die, that you can't talk to me openly about my cancer.
Understand that we want the truth.
My daughter's cancer progressed and they didn't tell us what to expect.
If I'm gonna die, I want my provider to tell me that.
we would have done things a lot different. We would have done more and played more.
AYAs want to connect with each other.
Ask me about feeling alone.
Nicole was 16, but she was treated with older patients on the adult floor, and there was no one anywhere close to her age.
It would have been so great for me if I had a friend like Tony when I was sick, and that we could have talked about things.
When I go for chemo, I don't want everyone in the room to be at least 60 years old.
And put us together.
Yeah, definitely.
If there's ways to schedule AYAs so that they can sit next to each other, I feel like that's such a huge thing.
You think in your mind that there are two people that just need to meet each other. Their situations are similar and they would really hit it off. This is done really easily by having both patients fill out a release of information form. Sharing that information allows the two patients to connect, and there's no issue with HIPAA.
We want you to bring as many things to the table as you can. Nutrition and acupuncture, exercise, anything you can think of, we would want to know.
Exercising helps me feel like a normal young adult. I want to work out, and I want to do it in a way that's safe.
When they told me I had 6 months to live, I learned through my dietitians what the best foods to put into my body.
Well, this one's awkward for a dad, but you need to talk to us about sex.
From the first day.
And remember to tell me about fertility preservation.
How or if we're going to be impacted.
During my treatment alone, the sex talk never came up because I was being treated inside a children's clinic.
He also asked me in front of my mom if I was. I wish I had been asked privately.
Please don't assume that I'm not interested because I'm in the middle of treatment.
And if I say that I'm really not interested ever again, maybe refer me to someone to talk to about that.
For your final, I would advise you to remember the following points.
Right. Thank you. Anyone have questions for Jonathan and me? If you need to leave, go ahead, but we can, Jonathan, do you have something to say? Yeah, come on up. That video is available for free on the Teen Cancer America website, and so we hope it will be useful for clinicians in all areas. And, you know, it is cancer specific, but there's a lot that actually could be true for all AYAs. All right. Thank you so much for coming. There should be a sign-in sheet, so if you haven't signed it yet, please. Oh, great. Made it all the way around. Okay. Take care. Everyone online, thanks for joining too.
Thank you. Bye.